Thank you for the introduction, and I'd also like to thank the organizers for the invitation. As I was saying, I wish I could be out there, but we're doing the best we can given current circumstances. So what I would like to talk about today is how we can use structural interaction, particularly derived from molecular modeling, in order to figure out what proteins do and how they interact, as the title says, at genome-wide scales. And I'd like to begin just by giving an idea of why we are interested in these problems. And that is that in any organism that you look at, my lab is mainly interested in bacteria, but this is true everywhere. There are still a large fraction of proteins encoded in genomes where we do not know their function. And yet, despite this, in many cases, these proteins are important for fitness. And so to give an example of this, I like to look at E. coli K12, which is probably the best studied bacterium on the face of the planet. And despite decades of study, though, depending on how you count it, between 10 and 15% of the protein coding genes in this genome are basically unannotated. But this amounts to several hundred poorly annotated proteins in this very well-studied proteome. But what I think is important to note is that even though many of these proteins are unannotated, they are not unimportant. And so if, for example, you look across a meta-analysis of knockout experiments where people looked at the fitness effects of deleting genes, what you would see is that if you look at a breakdown of knockouts that were either beneficial to the cells or deleterious to the cells, the breakdown of these proteins that are unannotated is about 10% of the significant fitness effects were on poorly annotated proteins. And so this is proportional to their representation in the proteome. And if you look at specific genes, you can find some genes that actually are poorly annotated, but have a very large number of conditions where they make a difference. So this is a analysis of 144 different knockout conditions. Here's a gene, for example, YCIB, that was problematic for the cells under 16 conditions to knock it out. Here's a gene YIBA that was helpful to knock out in 5 conditions. And so we see that these poorly annotated proteins can still be important. And so one of the things that we are really interested in is figuring out what poorly annotated proteins do as efficiently as possible. And this sort of ties into a larger difficulty that modern biology faces. I call this a abundant data problem. And problem is in air quotes because this is not really a bad thing that we have so much data, but it underscores the challenge. So if we look at the Tremble database, which contains a collection of all of the known protein sequences that are available, for example, from genome-wide sequencing at this point, what we can see is there's been exponential growth over the last few years of the size of this database. And this very much corresponds with advances in sequencing technology. You'll note that there's a downward blip here where there was a lot of redundancy pruning that happened in this database, which bought a couple of years of growth, and now it continues to rise fairly rapidly. Now, at the same time as known protein sequence space is expanding quite rapidly, if we look at the SwissProt database, which contains curated annotations of proteins, this is flattened out at the same time, which is really problematic. This is not the way you want this curve to look, because at the same time as we are knowing so many protein sequences, we seem to be decreasing our efforts to actually figure out what they do. And to even get a full impression for how different these are, I'd like you to note the y-axes. You have to plot these in a log scale together to have any comprehensible way of plotting them together. And the bottom line is only a few percent of protein space is in this well-annotated SwissProt database. So clearly the numbers of proteins that are involved here are gigantic. We need to figure out some way to annotate them. And I also want to point out that if we look at how large this poorly annotated protein problem is in important species, if you look at humans or E. coli, which are probably the 2 best studied genomes on the planet in terms of protein annotation, you're in the range of 10 or 15% of the protein coding genes being poorly annotated. If you look at other important model organisms, for example, yeast S. cerevisiae or the reduced genome mycoplasma, JCVI SYN3, you're in the range of 25 to 30% poorly annotated proteins. And then when we move on to important pathogens like C. difficile or M. tuberculosis, you're at about 50% poorly annotated proteins. So this is clearly a substantial problem. And then when we move to, say, rice, which is extremely important for human health and nutrition, you're in the ballpark of 90% poorly annotated proteins. And so really, these are important genomes where we do not know the functions of a large chunk of the proteins, and that reduces our ability either to engineer them in some cases or to identify potential drug targets in others. So one would think then that we would have an efficient computational method for taking a protein sequence and identifying what its function is going to be. It turns out it's not quite that simple. And in particular, pipelines for protein function prediction that are grounded in sequence information, which is the most easy thing that we get out of genome-wide sequencing, I'll add, or any kind of transcriptomic sequencing, for example. These are known to become less and less reliable as the amino acid sequence identity between a protein and its nearest well-annotated homologue drops below 60%. So the challenge for this field is if you look at the distribution across all protein space of the sequence identity between a given protein and its nearest well-annotated homologue, this distribution peaks around 30% sequence identity, which is well below the range in which purely sequence-based approaches have been shown to be reliable. And in fact, it's only 10 or 15% of protein space that's really in the easily annotated range, at least using purely sequence-based approaches. So somehow we need to pull in more information, and we need to have a computational workflow to do this annotation simply because protein space is too vast in order to be able to do anything else. So working together with Yang Zhang's lab, my lab has recently been working with a pipeline called Cofactor that we use for blind structure and function prediction of proteins. I'll explain why both of these are important. In particular here, we're emphasizing the function prediction. We would really like to understand what these proteins do. And so if you look at how Cofactor works, it is a hybrid pipeline that begins from a query sequence, so just an amino acid sequence that you might get, for example, by translating a ORF that you find in a sequencing experiment. And then there are three component pipelines of Cofactor. The first is purely sequence-based. We do sequence alignments. We assume that the unannotated protein probably does something like the nearest similar sequences. And so this is very similar to what other people do in this field. We also go into protein-protein interaction databases, and we look at the interaction partners of similar proteins to our query proteins, and we try to pull functional information from this. But then I think the most interesting part of this pipeline is we also end up doing a structure prediction, and we use structure alignments, and I'll go into a little more detail on this in a couple of slides, but we use structural alignments to find similar protein structures and assume that our query protein does something similar to the most structurally similar proteins. And so the key insight driving this approach is that it is structure and not sequence that is really going to be giving a protein its function. And so we should be paying attention to the structure when we're trying to figure out what a protein does. And so especially because this workshop is a lot on physics, I just wanted as a little bit of an aside to go into this structure prediction stage. I'll point out this is my collaborator Yang Zhang's lab that does this stage more than my lab working on it. But I think it's important to think about how do we make this jump from the sequence to the structure. And so the key approaches that are being used here, again, methods developed in Yang Zhang's lab. The first is a program called I-TASSER for template-based structure prediction. The idea behind I-TASSER is that it can be used whenever there are reasonably identifiable threading templates, which are proteins that are at least similar enough in sequence. They don't have to be very similar, but similar enough in sequence and in likely structure that you can take your query sequence, you can come up with a set of structures for at least components of that protein based on threading on existing structures. And then you can do a couple of rounds of refinement where first there's a reduced potential that uses just C alpha and C beta atoms that's used for initial rounds of structure refinement based on an initial pool of models. You then cluster the refined models, find the most likely structure based on center identified in the clustering, and then do some additional refinement, again, using this knowledge-based potential on a C alpha, C beta model, and ultimately do some all-atom refinement to get a structure. And so this is very much informed by a combination of physical and statistical potentials. Now, this works all right as long as there are identifiable threading templates that can be found. In cases where you have a query protein that does not have any good threading templates, you instead have to do ab initio protein structure prediction. And in this case, we use a program called Quark. So the idea behind Quark is that in the absence of good threading templates to figure out what the fold of a protein is, we're going to assemble an initial structure for a protein based on small fragments of known structures that are expected to be similar to the query sequence, just based on sequence similarity. And then there's going to be a set of replica exchange Monte Carlo refinement that's done on this. And this uses a bunch of rigid body moves that are demonstrated here, and also torsional rotations to allow efficient sampling of the space. And in this case, the representation of the protein is a little more complicated. You have all of the heavy atoms of the backbone plus the side chain centroid. Again, this uses a combination of a physical and a statistical potential to get you a set of models that are ultimately clustered and used to get an all-atom structure. And then for both this template-based and free modeling approach, over the last couple of years, they've been augmented by deep learning-based contact map predictions. And so the idea here is, while the physical and statistical potentials that are used in the modeling can do a pretty good job of predicting structure, giving them information based primarily on coevolution evidence between the different residues in proteins on where there are contacts can substantially improve performance. And so the idea behind these methods in general is you take a database of aligned sequences that are related to a query sequence and can identify where there is covariation or a shared information between mutations at different sites on the protein across evolutionary timescales. So then there's the assumption that that coevolution information within a single protein chain across evolution tells you what residues are in contact with each other. And then one can feed this into a set of neural networks that predict contact maps, which are then used as restraints in building and refining the atomistic structures. And so if I can just say one other word on the structure prediction problem, I think it's over the last couple of weeks, we've seen the release of the Cas14 results. And what really impressed everybody in the field, I think reasonably so, is if you look at the performance of groups across the open competition in Cas14, which was just released recently, you can see one group clearly performing well above the rest, and this was the AlphaFold group, and this has gotten a lot of attention recently. I think one important aspect that I would like to point out that has been less well covered is if you look only at the server groups, so these are only groups that have fully automated methods that they're willing to release, the 4 out of the top 5 for, 4 out of the top performing 5 server prediction sets came from the lab of Yang Zhang, my collaborator, from whom we get these methods. And so this is the I-TASSER method, this is the Quark method. And so I think in general, while the AlphaFold performance is extremely impressive, until we see automated servers based on these methods, I think it's a little premature to want to jump purely to deep learning-based methods and abandon all of the physical and statistical potentials that we use for these predictions. So in any case, that's enough of an aside. I'd like to get back to the structure prediction at this point. And so as I was saying, we're really building a lot of this prediction method around the fact that structure rather than sequence dictates protein function. And so we use this combination of sequence, protein-protein interaction, and then importantly, structure similarities in order to predict the functions of proteins. I'll zoom into the way we use that structure-based information in a minute, but I want to say also that one of the interesting things we did when we started developing this method was we asked, are current structure modeling methods good enough for us to be able to identify protein functions? And the way we did this was we used structural alignments to predict protein function, either in the case where we used only crystal structures, so we knew what those things did, or what those things looked like, or where we used predicted structures where we imposed a pretty stringent threshold on not allowing any structural information from similar structures to inform our modeling. And what we found that the modeled structures performed as well as the crystal structures for the purposes of functional annotation. So that does not mean that those models are perfect by any means. They're probably not. In fact, I know that they're not. But in general, we are good enough on the modeling side to identify similar structures for the purpose of function prediction. And so when you pull all of this information together, the structure information enters our predictions in two different ways. First of all, we take our query structure, we do global alignments against the entire protein data bank, and then we pull functional annotations from things with similar global folds. We also identify using a couple other methods. They're detailed in the cofactor paper. I don't really have time to get into these. We identify likely functional residues just based on where there are cavities in the protein. And we do local structural alignments within those likely binding sites and pull additional information both on similar proteins as proteins that have similar possible binding or active sites. And from that, we also pull information on likely molecules that may bind to those proteins. So when you pull this all together, we get a set of predictions that look like this. This is the web page for cofactor. And so we get both a set of similar proteins that are identified. These are proteins with good structural similarity to our query structure. And very frequently, they don't have high sequence identity. So here, for example, these are 3 to 4 angstrom C alpha RMSDs, but the sequence identities are below 15%. And then we also get a set of protein function predictions. And so these function predictions come in the form of directed acyclic graphs because they are Go terms. The closer to red, the higher the confidence in the predicted function. So here, for example, we have a protein where we're very confident in saying it's some kind of hydrolase, and at least have some confidence that it's particularly acting on carboxylic esters. So that's enough with methods. I think it's important to see how this thing actually does. And so to ask that question, we did what was called a re-annotation task on E. coli K12 proteins, where we took a set of a couple thousand proteins from E. coli K12 where we know their function, we pretend we don't, and then we try to repredict those functions. And so what I'm showing here, there's a performance metric called the Fmax, the maximum F measure, on the y-axis. And then the x-axis is difficulty, whereas we go from right to left. We mask from our databases all the proteins that have at least that high a sequence identity to the query protein, and then we look at how well we can do. And so the yellow and cyan lines are performance using BLAST or SciBLAST, where we just look for similar sequences to our query protein. This would be the purely sequence-based approach, and then assume that our query protein does something similar to the most similar sequences. So if we look at the performance of BLAST and SciBLAST, they do fine if we have high sequence identity targets to annotate based on, but they do worse and worse as we have lower and lower sequence identity to annotated proteins. And when we get down to about 30% sequence identity, we drop below this gray line, which is you get just by guessing Go terms based on their abundance in UniProt. Now this, on the other hand, is the performance just of the structure-based pipeline from cofactor. And what I think is very important to note here is there is very little slope to this line on the basis of sequence identity. There is a slope here, it's just very small. And so what this tells us is that indeed structure information carries robust information about function across long evolutionary distances, which lets us get good robustness and good predictions even on proteins where we don't have any good sequence identity templates to base our functional predictions on. And so I can also add in the blue here is the sequence-based pipeline from cofactor, which does pretty well, but it does decay in performance when you have low identity targets to annotate from. The green is the protein-protein interaction-based pipeline. And you put all these together and you get the hybrid model of cofactor, which is in the black line, which gives you sort of the best of both worlds, where we have very high performance when there's high sequence identity available, and then very little decay as you go to low sequence identity to the nearest annotated homologs being available. But of course, this is a bulk set of predictions, and I have experimentalists in my lab. They're interested in detail. So it's good for us to look at a couple cases where we actually have performed these predictions. And so just as one case study, I'd like to show the protein YBHP from E. coli. This is the UniProt page on YBHP, at least from a couple of years ago. And so it is listed just as uncharacterized protein YBHP, and that is literally all of the information that we have. But we know we're interested in this protein because if you knock it out, it has a profound impact on antibiotic resistance. And so the first step in figuring out why is figuring out what this thing does. So in contrast with this not very useful uniprot annotation, if we do our structure function prediction pipeline, cofactor, this is the set of functions that we get, and here is a predicted structure. And what we can read off of these predictions is this thing is expected to be an endonuclease. That is, first of all, the identity of the highest confidence leaf node in this graph. And we can see why this prediction comes up pretty easily, that we have here the predicted structure of YBHP, and it has, based on structural similarity, a predicted binding site for double-stranded DNA. And so one would immediately predict that this thing is going to be an endonuclease acting on double-stranded DNA. And sure enough, if we actually purify YBHP and we give it DNA in the presence of divalent cations, we can see that with increasing amounts of YBHP, here's the highest amount, YBHP plus divalent cation will lead to digestion of this DNA. So this is clearly acting as an endonuclease on DNA. And in particular, we can check the endonuclease function in a plasmid supercoiling relaxation assay. And so to do this, we take some supercoiled plasmid here. And if this has endonuclease activity, it should be able to nick or break that plasmid and lead to relaxation of the fully supercoiled band, which is here. And sure enough, we treat with YBHP in the presence of manganese, and you see relaxation of the fully supercoiled band. We are also able to abolish this activity with 2 point mutants. So the orange boxes here are the same digestion experiment, but where we've mutated one of 2 key residues that we identified based on that predicted YBHP structure, and you can see a loss of activity. So we're able to go from we have no clue what this protein does to we think it is a endonuclease acting on double-stranded DNA, and here is the active site. And this is the kind of information we can get from these predictions. For the sake of time, I'm going to skip a second example, so if you'll pardon me for a minute. So the point we've gotten to, now that we have this reliable structure-based functional annotation pipeline, we can move to the level of entire proteomes and start to ask questions of what are all the structures in these proteomes, and then what are the likely functions, and in particular, what are the likely functions of genes that we don't already know a lot about. And so we've done this, first of all, with the E. coli K12 proteome that's shown here. So we take about 4,200 protein sequences. We predict structures based on either I-TASSER or Quark, depending on whether we need a template-based or a free model. And then we put these into our cofactor and also a refined pipeline called Metago for doing function prediction. And when we do this, we get then function and structure predictions for a large number of proteins. We call this the E. coli Protein Information Collection, or EPIC. This is going to be going up on a website soon. And so overall, this took about 600,000 core hours of computer time for the modeling plus the function prediction. But what is immensely useful here is we get these proteome-wide predictions of both structure and, importantly, function. And so then we were really interested in looking at what classes of function were specifically enriched among the current poorly annotated proteins. In essence, we want to know what we're missing from current functional databases. And so what we do here in this volcano plot is we test the abundance of different gene ontology terms, or GO terms, which define protein function, among poorly annotated versus well-annotated proteins. So the further to the right you are on this plot, a given GO term is more enriched in the poorly annotated set of proteins. Things to the left are more enriched in the well-annotated set of proteins. The log-fold enrichment is on the x-axis. The p-value for that enrichment is on the y-axis. And so, and then the size of each dot corresponds to the number of proteins in the E. coli K12 proteome that has that particular GO term. And so if we look, first of all, at the things enriched among the well-annotated set of proteins, this is the GO terms over here, it's rather reassuring to see that we see things like central carbon metabolism, ribosomal components, things that we know roughly what they do among this set. Not surprising. Now, the interesting side of things comes if we look at GO terms that are enriched among the poorly annotated set of proteins. And when we do this, so these are the GO terms over here, we see enrichments of things like phage components, likely coming from prophages in the genome, things involved in particular nucleotide metabolism, enzyme activators, and the highest enrichment is for proteins involved in pathogenesis. And so this is very interesting because we are not only identifying unknown protein functions, but we're finding things that are probably involved in pathogenesis in these proteome-wide screens. And so just to give a couple examples of proteins in those categories, one I want to highlight is one protein called YJBI. The uniprot and ecopsych annotations to this protein are that it is some kind of conserved protein, but give no notion as to the function. Now, our cofactor predictions on the other side suggest that it's involved in multi-organism processes, first of all, and pathogenesis, and then also that it in particular acts as a ubiquitin transferase. And this may be surprising to see in bacterium, but it's actually not surprising because ubiquitin transferases are injected into host cells by many pathogenic bacteria to perturb the host cell physiology. And we can see where this prediction comes from in this case, because if we look at our model of YJBI, which is shown in blue, you can see very good structural alignments to the ubiquitin ligases, or sorry, ubiquitin transferases, both from an E. coli, enterohemorrhagic E. coli in green, and also Salmonella typhimurium in red. And so what you see here is actually highly characteristic of what we see when we do these structure-based function annotations. There is low sequence identity between these proteins. It's between 14 and 19% sequence identity in these cases, but there are very good alpha carbon RMSDs, and that tells us that the structures are very similar, even though the sequences are far apart. Just to show one more example of this, there's a second protein called YDDB that was predicted in our study as a poorly annotated protein involved in pathogenesis. And in this case, here again, it's currently listed as an uncharacterized protein. We predicted that it's involved in iron uptake, which is very important during pathogenesis because there's a sort of a war for iron between the immune system and of the host and a pathogenic bacterium. Where this comes from is a predicted structure of this protein that shows very good structural similarity to a known iron transporter, FEPA. And actually, the crystal structure of this YDDB was recently solved and takes on this exact fold. Not obviously a perfect structure prediction, but it takes on this same beta barrel fold characteristic of an iron siderophore uptake protein. And so for the sake of time, it seems I'm running out of time. So I'm going to have to skip over my other set of examples, which involve similar applications to the JCVI SYN3 minimal genome mycoplasma. I'll simply say with this that we're able to go from having a large number of poorly annotated proteins among currently unannotated proteins in this proteome. This is the hardest category of targets. We're going from knowing about 5% of them with functions to 50% of them with functions after the structure-based functional annotation on this proteome. And we've recently moved also into adding structural information to sequence-based information and to sequence similarity in order to predict protein-protein interactions. This is currently in early developmental stages, but we're actually seeing very good performance of these methods compared with existing methods. I'll show this as one last slide and then move on to my conclusions. And so my apologies for flying through this. I guess it's early in the morning here, so I didn't talk as fast as usual. So PEPI is the pipeline that we've come up with for this protein-protein interaction prediction. And this is in particular looking for physical contacts rather than just functional contacts the way most pipelines would. These are the areas under the curve that you get for the ROC curves for predicting protein-protein interactions using this pipeline. On the other hand, PRISM is an existing method in the literature that uses an interface and docking-based approach that doesn't seem to do as well. So we think that we're able to get substantial improvements using our hybrid models on finding protein-protein interactions as well. So if I put together the insights that we get out of these studies so far, what we see is that, first of all, pulling in structural information gives substantial improvements both in the quantitative predictions that we get, or the quantitative performance of predictions in terms of getting Go terms right, and also the qualitative use of those function predictions in terms of giving experimentalists a good starting point. We see that current methods in protein structure prediction are good enough to be used in functional annotation, and that now we can get whole proteome function predictions in reasonable amounts of computer time. It gives us a really good starting point for experiments on the poorly annotated sets of proteins in these proteomes. And so I think the near-term opportunities we have for improvement that we're currently working on are things like better curating binding site data to get more information from that, having better methods for identifying the key residues so we can emphasize more of the alignment around key binding sites, and then a lot of expanding into whole proteome annotations in other species, and also pulling in the protein-protein interaction predictions that I briefly mentioned at the end. And then I think circling back to the very beginning, I talked at the very beginning of how molecular modeling informs these structure predictions. I think that one really important thing that we need to do as we start to get these very large databases of structure predictions and function predictions is to circle back to molecular modeling and use that to be able to make additional predictions on function and on docking. And you saw, I think, an example of this a couple of days ago in Helmut Grubmüller's talk, where he was talking about the sort of dynamome where you could use dynamic information to further inform function predictions. And that's something that's also enabled by having these large-scale structure function predictions. So with that, I'd like to thank the folks who were involved in this work. And these were Chenxing Zhang and Eric Bell, who are grad students who worked on the computational side, and Maiti Rahimpur in my lab, who did the experimental follow-ups, Yang Zhang, who's been my long-term collaborator on the structure function prediction pipelines, and of course, our funding sources. And I'd like to thank you all for your attention and be happy to take any questions that you have.
Thank you for this nice talk. Are there, oh, my chat disappeared. Yes, are there some questions? I would just start with a remark that I find quite reassuring that to hear that deep learning cannot do everything and that we still have to use our brain. It somehow justifies why we are paid.
Yeah, I think it can certainly do a lot. And I think there was a lot of heartburn when those Cas14 results first came out. And I think it is certainly becoming an increasingly valuable and powerful tool, but certainly not yet a substitute for, as you say, using our brains. I think it's also not a substitute for having some knowledge-based models for what is going on.
So if there are no other questions, I have one. I'm not familiar to this field, so it's quite naive. But at the beginning, you mentioned that for the human genome, we had 15% poorly annotated. And I was wondering, because I understand that well annotated is not well defined. You can always annotate more. But when it's poorly annotated, what do you mean? Do we know nothing?
Yeah.
Or?
No, I think that's a great question. And so this is a very hard thing to do, particularly in bulk. So what we are basing that poorly annotated qualifier on is in the UniProt database. They assign an annotation score from 1 to 5, with 5 being the highest. This is done by curation based on how much information is available either in the literature or on proteins with very similar sequences. And so we consider as poorly annotated things that are 1 or 2 on that UniProt scale. And the reason we picked that is by doing a lot of our own investigation and looking in practice at proteins from, say, E. coli, and asking, is the annotation in UniProt of any use if you wanted to figure out either what the molecular function of this thing is or what biological process it might be involved in? And the answer was anything with a 1 or 2 gave essentially no information. And there are actually even proteins with higher annotation scores where we don't know what they do or what physiological process they play a role in. For example, I have a grad student on an unrelated project who's working with a protein where we know roughly what function, what reaction it catalyzes in vitro. We still have no idea of its in vivo role. But here, we're focusing on the things where we truly have little or no information. So an annotation score of 2 might be, we have a pretty good idea it's a hydrolase of some sort, maybe a general enzymatic class. 1 is we really have no idea.
Okay, I think we don't have any other questions. If you have one, please raise hand or put in the chat. If not,